Hi, good afternoon everybody. Now for today's video, we'll be discussing gene segregation and integration. Since this video is just a continuation from Miss Taring's report, then let us now start. Modifier genes. Now, genes that affect the phenotypic or a molecular expression of other genes in a quantitative fashion. Genes that have small quantitative effects on the level of expression of another gene. Take note, it has only a small quantitative effects. Therefore, modifier genes, ito yung nagmo-modify ng ibang gene. Example, the coat color is refers to as the illustration as dominant B, which refers to black, and recessive B to brown. And ano yung intensity ng kulay niya? Either dominant D, which is full, or recessive D, dilute, or faded. That is not brown, sorry. Now, as an illustration, there are 9 dominant B and dominant D. Bakit black ang color niya? Kasi dominant B, which refers to black, and dominant D, which is full, means it is full black. At ito namang 3 dominant B, may tatlong 3 dominant B and 2 homozygous recessive D, which refers to dilute black. Kasi yung B, which is to black, and yung D, dilute or faded. Kaya, yun po ang illustration niya. So, ito yung modified genes, nagmo-modify lamang siya. There is always a connection between them. Okay. Next, we have the illustration of modifier genes. Like what I have said, there are 9. Then the ratio there is 9:3:3:1. Okay. Next, we have the gene interactions. Remember, there are some examples of there are concept of gene interactions, the novel phenotypes. There is a complete dominance in both gene pairs. It is has a result new unique appearance. The novel phenotypes always has a new unique appearance. If the phenotypes result from interaction between dominants, interaction between both homozygous recessives, may interaction sa dominante, may interaction sa homozygous recessives. Example, the comb shape of a roaster. The interaction there is that the P and R, the dominant P and R produces walnut comb, and that walnut comb, which refers to the offspring, and the example there is the walnut comb is ibang-iba siya sa parents niya. Therefore, the interaction of the dominant R and the dominant P produces walnut. At yung novel phenotypes naman, nagpo-produce naman siya ng homozygous recessive R and homozygous recessive P, which is single. Kaiba naman ito sa appearance ng kanyang parents. That is the novel phenotypes. So, now, the illustration, the same, the concept there, the same, that yung blue color nagre-represent siya sa walnut, yung orange naman nagre-represent sa rose, yung pink color nagre-represent siya sa P parents, which is ang color naman ng green nagre-represent sa single. So, we have 9 walnut, 9 rose, 3 P, and 1 single. That is the novel phenotypes. Second, the recessive epistasis. Again, highlight ko lang po, one gene, the recessive epistasis is that one gene when homozygous recessive hides or masks the effect of the other. But before tackling that, move to look at the right side. What does epistasis means? It is a type of polygenic interaction where one gene controls the phenotype of another gene. In short, masking. Parang tinatakpan niya yung kulay ng ibang gene at siya yung makikita. Now, the recessive epistasis is that yung mga maliliit na letra, which is recessive, siya yung may kakayahan na magtakip ng ibang kulay. O yung example, albino, the one, mayroon tayong homozygous recessive A at homozygous recessive C. O, di ba? Nagpo-produce siya ng albino. Makikita natin yan dito sa kanyang parents, which is the homozygous recessive C. May homozygous recessive C, which is albino. So, the interaction is that the homozygous recessive C is epistatic to agouti and black. Kaya, the concept there is recessive epistasis. Siya yung may kakayahang magtakip ng ibang kulay, magpakita ng kakayahan. Now, the same illustration lang po dyan sa Punnett square na makikita ninyo. The ratio there is that 9:3:4. Bakit nagiging 4? Kasi as you can see, there are 2 albino there, which is may tatlo at isa. Now, we will add that, and that is why we get 4. 9:3 is to 4 yung ratio natin. So, dominant epistasis. Sensory recessive epistasis, kaibahan naman ito sa dominant epistasis. Ang dominant epistasis, siya yung may kakayahan na magbigay ng kulay o ng phenotypic expression ng isang gene. Yung dominante. When one gene, when dominant, hides or masks the effect of other gene. That is why dominant epistasis. Example, yung 9 dominant B, dominant Y and dominant W. Kaya yung W nagtatakip ng kulay sa white kasi dominant po siya. Though yellow is also dominant, but siya yung unang dominante. Now, we have three dominant W and homozygous recessive Y, kaya nagiging white. Basta, the concept there, when dominant, it hides or masks the other effect. Now, the three dominant epistasis is the same concept lang po siya sa dominant epistasis. When dominant, it has a capability to hide the other effects of other genes. That is dominant epistasis. Now, complementary genes. The rule there is that either recessive homozygote is epistatic to the effects of other gene. Gene interaction between two different genes that work together. That is complementary. Example here is that kapag makikita mo na may homozygous recessive siya yung may kakayahan. Parang recessive epistasis na rin siya, pero iba itong complementary. Since the genes P and C produces enzymes that catalyze successive steps in a chemical process leading to production of pigments. Nagpo-produce ito ng enzymes na nakapagbibigay ng ibang kulay. Halimbawa, the gene P nagpo-produce ng enzyme P and the gene C nagpo-produce ng enzyme C. Now, the precursor there is that the produces white color and purple color naman yung pinoproduce ng gene C. Kaya yung white, nabibigyan ng ibang chemical pigments ng gene C or ng enzyme C, kaya nagiging purple yung kulay. There is a complementary result. Now, the complementary gene, the same concept lang po siya. Bakit yung ratio niya is 9:7? May tatlo tayong white. We will add those whites. Kaya 3 plus 3 plus 1 is equal to 7. Kaya 9:7 yung ratio natin. Now, the duplicate genes naman, the concept there, either gene, okay, we will zoom, either gene when dominant is epistatic to the other. Again, if it is dominant, it will hide the effect of other. Example, the 9, there is a 9 dominant A and dominant B, which is referred to triangular. And kahit na may dalawa tayong homozygous recessive B, still the offspring or the product is triangular kasi yung dominant A, siya yung nagpapakita ng kakayahan na magtakip ng ibang kulay. Dominant B, again, triangular. Look at the parents. Diba? They are heterozygous dominants. Now, the effect is their ovoid kasi heterozygous recessive sila. May homozygous recessive A, may homozygous recessive B. The interaction there is that a dominant allele at either, what is it? At the dominant allele, either the dominant allele either recessive hides the effect of the ovoid. When dominant, it hides the effect of the ovoid. Now, the same concept, pseudoalleles. What is pseudoalleles? It is a closely linked genes having similar effects. Two genes with similar functions located so close to one another on a chromosome. Kapag dalawang gene na matatagpuan sa isang chromosome na duol na kayo sila sa usag-usa. They are closely linked genes. They are closely to one another. Napakalapit lang nila sa isa't isa. That is pseudoalleles. Now, environmental influence on gene expression, we have two factors, external and internal environmental. Under the external environmental, like foods, drugs, or exposure to toxins can cause epigenetic changes by altering the way molecules bind to DNA. Like external environment, yung nakipag mga Example, temperature and or light will cause and influence gene expression. Example, Himalayan rabbits carry the C gene, which is required for the development of pigments in the fur, skin, eyes, and whose expression is regulated by temperature. Okay, that is the external. Internal naman, like hormones and metabolism also affects gene expression. Example, gender can also affect too, as it is the case with sex influence with sex limited traits. Example of sex influence trait is the male pattern baldness. The baldness allele which causes hair loss is influenced by hormones. Yung pagkakalbo ng tao. Kaya effect yun ng ating internal environment sa ating katawan. Now, the study of the twin studies, the concordance and discordance. Ang ibig sabihin is that the concordance, presence of the same trait in both members of pair of twins. Twins are concordant when both have lack both have or lack of given trait. Kapag silang dalawa ang may traits, means they are concordant. Kapag sila dalawa ay may magkaibang traits, ang isa ay wala, ang isa naman meron, means they are discordant. Example, if in a twin pair, if only one twin in a pair has disease morbus, then the twins are said to be discordant. But if twins in a pair have the same disease, they are said to be concordant of morbus. Kapag isa sa kanila ay may disease lamang ng morbus at ang isa naman ay wala, means they are discordant. Kapag silang dalawa ay mayroong disease, traits na morbus, means they are concordant. There is a formula also, but if you have any clarifications about the formula, you can ask it on our group chat. Now, monozygotic is refers to identical and dizygotic means fraternal. Monozygotic is that twins have the same genes. Twins result in a single egg fertilized by single sperm. Mono meaning isa lang. Dapat dizygotic or fraternal means they share 50% of their genes. Twins arise when two separate eggs are fertilized by different sperms. Kapag dalawa, meaning dalawa. So, halimbawa, kung yung discordant twins, silang dalawa ay concordant kasi mayroon silang parehong traits. Isa sa kanila ay kapag isa sa kanila ay walang traits, ang isa naman meron, meaning they are discordant. Kapag dalawa sila mayroong traits na iyon, they are concordant. Now, we will move on to probability and statistical testing. Hypothesis testing is a form of statistical inference that uses data from a sample to draw conclusions about a particular parameter. What does probability means? The chance that an event will occur expressed as a fraction or percentage. Now, we will move into chi-square test. The formula of chi-square test is that chi squared is equal to observed minus expected squared over expected. Now, the rule is that if the chi-square value is greater than the value at a specific probability, then the null hypothesis has been rejected and a significant deviation from predicted values was observed. Null hypothesis will be accepted if the result is less than. Therefore, kung matapos nating isolve ang chi-square na yon, matapos nating isolve yung mga classes, yung observed and expected variables natin, kapag ang resulta is mas malaki doon sa expected value natin, or meaning the null hypothesis should be rejected. If maliit ang resulta natin sa pag-solve natin, means the null hypothesis will be accepted. Now, the null hypothesis is that the main concept is that if, example, there is no significant difference between the observed variables and the expected variables, or there are no significant differences about the deviations or etc. Yun yung null hypothesis natin. Now, example, after solving is that we have 4 classes. Now, remember that df, or the degree of freedom, is equal to n minus 1, where n is the number of classes. May 4 tayong classes. Yellow, round, and so on and so forth. So, 4 minus 1 is equal to 3. So, now, observe 328, 96, 99, 37 is equal to 560. Now, paano natin kinuha yung expected? Now, so, The ratio there is that 9 is to 3 is to 3 is to 1. Yan pong ratio natin. Kaya 9 times 560 is equal to 5,040 over 16 is equal to 315. 3 times 560 is equal to 1,680 divided by 16 is equal to 105. Kaya and so on and so forth. Now, we will subtract 328 minus 315 is equal to 13. 96 divided by minus 105 is equal to negative 9 and so on. Now, we will get the squared. 13 times 13 is 169. Negative 9 times negative 9 is 81 and so on. Do not multiply 13 times 2 because that is not correct. Now, we will also move on to 169 divided by the expected 350 and the result is 0.536. 81 divided by 105, 0.771. 36 divided by 105, 0.343 and so on. And then we will add that column and the result is chi-squared is equal to 1.907. Since our chi-squared value is 1.907, we will move on, we'll observe it into the table. Now, yung main reference natin is 0.05. Kanina, the degree of freedom is 4 minus 1 is equal to 3. Therefore, sa 3 tayo dito sa DF. So, 3 o 0.05 meaning 7.82. Now, our result kanina is 1.907. So, since our main reference is that 7.82, mas maliit yung 1.907. Kaya, we will accept the null hypothesis that there is no significant differences between those variables that we observe. I hope that is right, and I hope you understand our report. And that is all. Thank you for watching.